Hi everyone, welcome back to Neurobiology at Providence College. I'm Joe DeGeorgis. Today we're going to review a paper by Adler and colleagues titled Alien Intracellular Calcium Chelators Attenuate Neurotransmitter Release at the Squid Giant Synapse. It was published in the Journal of Neuroscience in June of 1991. For this paper, the title really tells it all. A calcium chelator is a substance that binds calcium. They say that it's alien, so it's an external source that's placed into the intracellular compartment of the squid giant synapse, and that attenuates, inhibits neurotransmitter release. So a substance that binds calcium injected into the squid giant synapse stops neurotransmitter release. Pretty simple. Really cool paper. They say at the beginning of the introduction, many lines of evidence suggest that the release of neurotransmitter is triggered by a brief increase in the intracellular calcium concentration of the presynaptic nerve terminal. One prediction of this calcium hypothesis of neurotransmitter release is that an increase in the intracellular calcium buffering capacity should dampen this transient rise in free calcium concentration and hence attenuate neurotransmitter release. However, Adams et al. Have reported that injection of the calcium chelator EGTA into the presynaptic nerve terminal of the squid giant synapse did not reduce the evoked transmitter release. So let's talk about this a little bit. Like Riccardo Miledi, Adler and colleagues were using the squid giant synapse, and we've said many times before that the body of the squid is shaped like an ice cream cone, and it's hollow inside, and of course it has a head and tentacles with a big eye, and behind the eye are a number of lobes that make up essentially the brain of the squid. And a neuron comes down from the brain, two of them, actually, one on each side of the squid, and it connects to a series of giant axons that innervate this muscle, the ice cream cone itself, that they call the mantle. And the squid can take water into the ice cream cone and swell so that this gets a little bit bigger, like this. And then when this neuron fires, it triggers a coordinated firing of all of these downstream giant axons that cause the muscle to contract, and water is pushed out of the body in this direction, and then the squid jets forward in that direction, tail first. And we also said that you can take a pair of scissors and just simply cut the bottom edge of the ice cream cone, of the mantle, and you can then fold out the ice cream cone like this, flatten it out, and you have a big triangle. And of course there are organs in here, but they're very easily dissected out, and what you're left with is this piece of muscle, the mantle itself, and neurons that come down from that squid brain like this, one on each side, because the organism is symmetric. And I'm just going to draw one of the giant axons which connects like this. And there's, of course, one on each side, like that. And then this bifurcates and innervates the muscle and creates a neuromuscular junction. So what Adler and colleagues are really focused on is this synapse between this upstream neuron and the downstream squid giant axon. And just like Miledi, they put an electrode in the presynaptic terminal, that is the upstream neuron, that they can use to monitor an action potential on an oscilloscope. Can measure the action potential. And then they have a second electrode here that they use to stimulate this upstream nerve cell. So if you run a little electricity down this electrode, it will cause an action potential in this neuron that can be detected here by this electrode. And then they have a third electrode which measures an action potential in the downstream in the squid giant axon, like this. And so if you fire this and you detect an action potential in the first neuron, and then you detect an action potential in the second neuron, then neurotransmission has indeed occurred. Previously, we said that work by Riccardo Miledi and others demonstrated that if you remove calcium from the external media, that you inhibit synaptic transmission or neurotransmitter release. So usually this is done by adding a substance called EGTA, which they talk about in this paper. And EGTA is a calcium chelator. So it binds calcium and in doing so inhibits the effect of calcium on synaptic transmission. So this was a good indicator that calcium was probably involved in the process, and that the source of the calcium was calcium in the external fluid. Then, in the paper that we reviewed by Miledi used this electrode to microinject calcium directly into the presynaptic terminal, and that caused an action potential in the downstream neuron. And so the injection of calcium caused synaptic transmission to occur. It caused the release of the neurotransmitter. And that is without stimulating with this neuron. And in fact, we said that they poisoned this upstream cell with pufferfish toxin that binds to sodium channels and inhibits the action potential in this neuron from occurring. In today's paper by Adler, they wanted to further investigate the calcium hypothesis. The idea was that if indeed calcium causes synaptic transmission or the release of the neurotransmitter, then a calcium chelator like EGTA injected into the presynaptic terminal should inhibit neurotransmitter release. Once again, I'd like to draw the synapse at a much higher scale. In figure 1, they show a schematic of the squid giant synapse. It looks different than the synapse that I drew the other day for the Miledi paper. I drew a mammalian synapse in that particular figure. So I'll redraw their figure 1. This is the presynaptic cell, and it has these fingers that they show in the schematic, like this. So this is the presynaptic neuron, the one that comes down from the brain. And the reason it has all of these little fingers is because these fingers synapse with the many giant axons that I drew in the squid, like this. So these are the smaller giant axons, and then the largest of the squid giant axons is like this. So this is the synaptic connection here, and they call this the pre. Of course, this is the presynaptic terminal of the synapse, and this is the postsynaptic terminal of the synapse. And they would put an electrode, which is in the presynaptic terminal. They would put an electrode here, which would go to the oscilloscope to measure the action potential of the downstream axon to determine whether synaptic transmission occurred or whether the neurotransmitter was released. And then, of course, they put a stimulating electrode here in the presynaptic terminal. This electrode can do two things. It can be used for microinjection, and it can also be used to measure the action potential of the presynaptic neuron, like this. Okay. So in the calcium hypothesis, the idea is that if an action potential is propagated down the upstream neuron from the brain, and the electricity hits this end here, that calcium somehow enters the cell from the external media and triggers neurotransmitter release, which diffuses across the synaptic cleft, that is the gap between the pre- and postsynaptic terminals of the synapse, and that these neurotransmitters bind to receptors on the membrane and cause ions to flow into the cell. That is, it causes the resting potential to go towards zero from minus 70 millivolts, moves towards zero, and if it reaches the threshold, the sodium channels open in the membrane of the axon down here and cause the action potential to occur. So Adler thought that if you added a calcium chelator to this presynaptic terminal here, that when the calcium rushed in, it would be bound up by the calcium chelator, and that would inhibit the neurotransmitter from being released. They say, however, in the introduction, that Adams reported that injection of the calcium chelator, EGTA, into the presynaptic nerve terminal of the squid giant synapse did not reduce evoked transmitter release. So previously, someone did the experiment where they injected EGTA, a calcium chelator, into the presynaptic terminal, and it had no effect. If you fire this neuron, you still ended up getting a response in the downstream neuron. So they talked to a friend of mine, Rindy Jaffe, one of my former housemates and a very talented scientist, and she suggested using a different calcium chelator, something other than EGTA, and she suggested the use of BABTA. In order to make sure that they injected the BABTA and to estimate the concentration of BABTA in the presynaptic terminal, they made a fluorescent version of the molecule, and that's shown in figure 1 of the paper. The primary reason to try BABTA is it has a much faster binding kinetic than EGTA. So, by injecting BABTA into this presynaptic terminal, when this neuron was fired, you no longer received a signal in the postsynaptic terminal, even though, of course, you could detect an action potential in this presynaptic terminal. So I'm going to redraw this one more time. Okay, once again, here's our presynaptic terminal of the squid giant axon with the fingers. And then we have our giant axon here synapsing with this nerve cell. So this is the postsynaptic neuron, and this is the presynaptic neuron. We have our stimulating electrode. We have a recording electrode here that also was used for microinjection, which is connected to an oscilloscope that can detect an action potential if it happens in this cell. And then we have a recording electrode in the postsynaptic cell to determine whether synaptic transmission occurred. They use two different types of calcium chelators, one EGTA and one a fluorescent version of BABTA. It turns out that the EGTA didn't work when it was injected into this presynaptic terminal. That is, if you stimulate this nerve cell, you detect an action potential in the presynaptic nerve cell, and you also detect an action potential in the postsynaptic neuron. That is, synaptic transmission occurred even in the presence of EGTA. When they used a fluorescent version of BABTA, which allowed them to see the BABTA, that it was indeed injected into the presynaptic terminal, and based on the color, you could estimate concentration of BABTA. When this neuron was stimulated with this electrode, you could see an action potential in the presynaptic neuron, but you no longer detected an action potential in the downstream neuron, suggesting that the BABTA bound to calcium that was rushing in from the outside cell and inhibiting the effect of calcium on neurotransmitter release. We now know that there are voltage-sensitive calcium channels in the presynaptic terminal. So when the action potential runs down as a wave down the length of this nerve cell, the voltage-sensitive channel detects the action potential, it opens up, and it allows calcium to rush into the cell. Somehow, calcium then causes synaptic vesicles that are membrane-bound structures that are loaded with neurotransmitter to fuse with the cell membrane and release their contents, the neurotransmitter, into the synaptic cleft, which diffuses across this gap and binds to other receptors in the postsynaptic membrane that allow sodium to rush into this postsynaptic cell. And the influx of the sodium causes the cell to become less negative, and if it reaches the threshold, a action potential occurs. This experiment provided further support of the calcium hypothesis. What a wonderful set of experiments and a great paper. See you guys next time.